Okay, so the previous one quit, but it was at 15 minutes, so it might be good to have a break. So I want to talk now about the patterning that takes place in the um, egg chamber as development proceeds. So the first egg chamber that develops is the oocyte is at the posterior end, and we don't really know, unless there's been some developments that I'm not aware of in the last couple of years, but it's not clear what specifies the posterior as posterior. But once you have one egg chamber formed, then the anterior part of the older chamber signals to the younger egg chamber ahead of it to specify the posterior end. And there's a number of signals. We're just going to talk about a couple of them because if we went into too much detail, we could spend the whole semester on flies, and there's lots of other interesting. Flies are my favorite because that's what I have been studying, since before any of you were born, maybe your parents as well. But there's a ton of other really interesting organisms that we need to cover. So I'm going to try to keep it to just a few important signals. And so the signals actually lead to the formation of these stalk cells, and the stalk cells are like a very thin set of follicle cells that make a separation between the two egg chambers, and then there's signals kind of from these cells to specify these, and then there's signals in this direction, and then there's some interaction between the oocyte itself and the polar cells here. So we're going to talk about that. And then when this is specified, it can then send signals to the egg chamber ahead of it as well to let it know, okay, this is the posterior end. And everything then develops according to, posterior versus anterior and so on. Okay, so this is the details. I'm going to go through these signals. I'm going to go through these signals a little bit more in detail in the class, but I want to just have a quick run through. So if we look at the anterior part of an older egg chamber, the follicle cells, which are these ones, and then there will be ones here that are on top of, because remember the follicle cells are surrounding the whole egg chamber, and they signal with delta, and hopefully, well, you may or may not remember this, but delta is one of these membrane bound ligands, and that's delta, and then the receiving cell has the notch receptor. So delta and notch, that's not right, N. And then when these two bind, then there's a cleavage of the intracellular, the inside part of the receptor, and it goes into the nucleus and leads to the transcription of downstream genes. And so that signaling is taking place, and they're from the nerve cells that are at the front here, to the anterior follicle cells. And the two cells that are right at the very tip are called polar cells. And so, and then that induces the formation of the stalk, which is shown in green here. And so let's see, basically what's happening is there is a signal that's being sent now. It's called unpaired, and it's being sent to these cells, and it causes them to form a stalk. But the stalk, also there is signaling through these cells. And then that causes the cells at the posterior end of the chamber before to form polar cells, and that's shown over here. So these cells here are induced to form polar cells. And these polar cells then also secrete unpaired. So the UPD is the abbreviation for that. And that's a signal that's being secreted to the cells sort of in this region, and they are going to respond. I'm going to show you unpaired as part of the JAK-STAT signaling family, and so I'll show you the details of that in a minute. Also, if you look in the egg chamber of the younger the oocyte of the younger egg chamber, you will find that there is a lot of messenger RNA for gherkin, and gherkin is a signaling molecule too, and that's found sort of stuck in the very posterior-most region, along with the nucleus. That's the nucleus of that egg. And then when the unpaired signal is received, then that causes the gherkin messenger RNA to be translated in the oocyte. So now what you have is cells that have both unpaired protein, but they also have gherkin protein here. And so it is the combination. So it is signals from the unpaired and from the gherkin protein, and those combine to make posterior follicle cells. If you made a mutation and you actually eliminated the messenger RNA or the protein for gherkin, and you just had the unpaired signaling, you would have these cells. You would have cells like these that have the unpaired but not the gherkin, and they would take on the anterior fate. Okay, you have unpaired being expressed here and here due to those signals. So that's what the black arrow is here. That's the unpaired, and that's the unpaired. And when the unpaired is combined, the signal there is combined with the fact that gherkin is also a signaling molecule, and it's sending messages to these cells. They're getting two sets of information, and that makes them take on a posterior fate. If they only get the one set of information, then they know they're at either the anterior or posterior end, and without the gherkin protein, they take on that anterior fate. Hopefully that's clear. Okay, so I just want to go through quickly the unpaired signaling. So the receptor in the case of this one is called domeless. These are just, they have these funny names because they're named after how people saw the phenotypes when mutations were made. So there's the domeless receptor. Is present as a transmembrane protein. When unpaired binds to it, it causes dimerization. The two receptors come close to each other because that's the only way they can properly bind the ligand. And when they do that, this protein called hopscotch, which is also called JAK in some other Janus kinase, I think is the longer name. These kinases that are associated with the receptors are able to phosphorylate and activate each other, and then they can bind to STAT, which is present as a monomer in the cytoplasm. When these the STAT protein gets phosphorylated here and here, it causes the proteins to form dimers, and when they form dimers, they're able to enter the nucleus and initiate transcription of downstream genes. So basically, this is what is going on in these cells here and these cells here. That's the unpaired signature that's happening, and there's certain sets of genes that are turned on to form the termini, the anterior and posterior ends of the the embryo. When gherkin is also present, that specifies that it will be posterior. And if gherkin isn't present, then it takes on a kind of a default, which is an anterior fate. Okay, and then I just point this out to you. This is a table from your textbook, Figure 219, and it basically shows you the different families of types of signaling molecules that are active in the fly. And we've talked about already DPP, for example. We talked about gherkin just now and delta just now. We'll be talking about wingless and hedgehog later on, and we just talked about unpaired. So the level at which they're described in the lectures is what you need to know. A table like this is useful, and I would suggest making sure you understand kind of how the process works, such that, you know, if we didn't have any gherkin protein, what would you expect to happen? You know, in terms of the polarization of the oocyte. Gherkin not only has that anterior-posterior role, but it also has a dorsal-ventral role. So keep that in mind. And some of these other things we're probably not going to cover. And branchless, it's really interesting because it's how the tracheal system develops. However, we don't have time to cover this one. So I'm putting big X here so that you won't spend time learning this one unless you need it for another course or something like that. Okay, so now we have this anterior-posterior specification. The follicle cells here know that they're posterior, and that leads to, through a variety of mechanisms, a change in the microtubule organization. And so what happens is the microtubules all kind of rearrange themselves, and they do so so that the plus ends of the microtubules are at the posterior end, and then the minus ends are along the anterior end. So don't forget there are these ring canals. Some of the microtubules actually also extend into the nerve cells. And so a lot of mRNAs, some of them may be made in the nerve cells, and they may, you know, make their way through these microtubules to where the microtubule organizing center is. But then, depending on which of the motor proteins they're associated with, some of them are hauled to the minus end of the microtubules, and that, for example, is bicoid, because that localizes it onto the anterior part of the egg chamber, which is going to be the anterior of the egg. And there's proteins that fix it to keep it glued to the anterior end. And then Oscar, which is responsible for localizing nanos, which you remember as the posterior morphogen, that's on a kinesin, and it's a plus end directed microtubule motor protein, so it moves to the anterior end, and then Oscar is localized here, and then nanos is also localized here. So that's kind of how those two are set up. And then you know what happens after fertilization. Once the egg is matured and so on, you get the protein gradients based on the fact that the nanos and the bicoid messenger RNAs start to get translated, and then they diffuse away to create those gradients. Okay, so there's more, and when you read through the text, you'll find that there are more details. If you understand it at this level, that will be enough for this class. So after that posterior specification takes place, this is when the oocyte, that's when the microtubules rearrange, and the oocyte makes its way up into what we would call the dorsal anterior portion of the embryo. And what happens is that there is a bunch of gherkin protein that moves along with it. And so you have a cloud of just the gherkin protein up around. That's what this stain is here. This is not the nucleus, this is the actual gherkin protein. And it's important to keep these two straight. So the gherkin does its job to specify the posterior, but then it moves to a different place and does another job, which is to specify the dorsal side of the embryo.